Okay, so welcome everyone to the launch of our online seminar series in evolution and ecology. So my name is Elizabeth Duxbury, and I am a postdoc in the research group of Alexei Maklikov at the University of East Anglia. So myself and our wonderful team of co-organisers in the groups of Judith Mank and Simone Imler at the Universities of British Columbia and of East Anglia are delighted by the fantastic lineup of upcoming speakers we have planned for you, and we are grateful for the staggering support from the research community for this initiative. It is our great pleasure to open the series today with Professor Molly Przeworski, Professor of Biological Sciences and Systems Biology from Columbia University in New York. Molly is a leading population geneticist who explores the intriguing questions of how and why variation in recombination and mutation rates are maintained in vertebrate populations, including humans and primates. Molly has won many prestigious awards, including the Rosalind Franklin Young Investigator Awards. Today, she will present her talk, An Evolutionary Perspective on Meiotic Recombination in Vertebrates. There will be a question and answer session with Molly directly after her talk, so please post your questions in the Questions for Seminar Speakers Slack channel and upvote questions you would like to hear. We aim to keep the whole session to 1 hour in length. So, without further ado, it is our pleasure to welcome Molly to present her talk. Thank you very much.
Thank you very much for the introduction. I'm going to disappear because I don't think you need to see me while I'm talking. But before I do, I just want to take the opportunity to thanking you both for inviting me, but most of all for putting together this really fantastic set of seminar speakers. So I'm really looking forward to the talks that follow. I'll be glad to be done with it and be able to focus on those. So thanks again for the fantastic initiative and for including me in this seminar series. So I'm gonna hide myself, there we go. Okay, so today I thought I was going to tell you about a line of work that's been going on in the lab now for about 15 years, in which we think about meiotic recombination as a phenotype, and we ask about the causes and consequences of its evolution in vertebrates. And so to this end, I'm going to be, except for the fact that let's see. There we go. So to this end, I'm going to be focusing on meiotic recombination. So the shuffling of DNA that occurs during meiosis and the process by which, as you know, parents transmit not one of their 2 chromosomes to their offspring, but a mosaic of their 2 chromosomes. And in all sexually reproducing organisms, this process of meiotic recombination is essential to the successful completion of meiosis, because that is what ensures the proper segregation of chromosomes into the daughter cells through one of the outcomes of recombination, namely crossovers. Now, in most sexually reproducing organisms, including all the ones I'm going to focus on today, so vertebrates, recombination also plays a second key role in the proper alignment of homologous chromosomes, the process that leads the 2 homologous chromosomes to find each other and to zipper up. And because of these essential roles, as you might imagine, errors in recombination are highly deleterious, leading either to aneuploidy or to chromosomal rearrangements when the alignment is improperly done, like deletions and chromosomal rearrangements. So in humans, for instance, errors in recombination are actually the leading cause of spontaneous miscarriage, and when not embryonic lethal, of severe developmental disabilities. And because of these essential roles, recombination is obviously highly constrained, and it's often sort of thought of as a somewhat immutable process, something that should be almost fixed by the selective constraints acting upon it. In addition, I think it's fair to say that as evolutionary biologists, we often think of recombination as sort of the input to the evolutionary process, for example, in generating new combinations of alleles on which natural selection can then act, and sort of conveniently think of it as a fixed parameter of the model that we at least implicitly have in mind for how populations evolve. In fact, however, recombination is not just an input to the evolutionary process, but itself an output. It's a phenotype. And it's a phenotype that evolves in almost every regard that one can think to characterize. So for instance, if you think about the probability of a recombination event occurring per base pair, that varies by orders of magnitude across taxa. And even when the recombination rate is pretty similar on average between 2 species, as is the case, for example, for these 2 species of Drosophila, Nonetheless, the distribution of events along the genome can differ markedly, for instance, in the degree of centromeric and telomeric suppression. And if you think about things not in terms of averages or megabase rates, but if you zoom down into what's going on at the scale of base pairs and you ask how heterogeneous are recombination landscapes, how much do rates vary from base pair to base pair, there too, the pattern differs markedly across species. So for instance, in flowering plants, in yeasts, in mammals, there are so-called recombination hotspots, regions of a kilobase, maybe 2 kilobases, that experience orders of magnitude more recombination than the background, than the sequence around them. In contrast, in Drosophila species and Caenorhabidopsis, at that scale of resolution, while there's variation at the megabase scale, at that scale of resolution, there's very little heterogeneity. And if you drill down even further to the subset of species that use recombination hotspots, say you look at different mammals, you see that sometimes the location of these hotspots differs markedly across mammals. So here, for instance, is a figure that we published a few years back now, in which we considered the degree of overlap of recombination hotspots between two closely related species, humans and chimpanzees. And what you can see here is that the degree of overlap that's observed by our operational definition is barely more than you would expect simply by chance if you placed hotspots at random along the genomes of those two species. And so a marked change in the location of hotspots, even over very close evolutionary distances. So all these examples just are meant to underscore that recombination is an evolving phenotype. And so that raises the question of why that is. How has genetic drift and how have natural selection pressures shaped the recombination machinery to bring about the diversity of processes that we see across the tree of life? So that's what I'm ultimately interested in. Today, I'm going to focus on these questions specifically as they pertain to recombination hotspot usage to discuss how recombination hotspots are specified in vertebrates and something about how and why they evolve so quickly. So the story I'm going to tell you starts with the publication back-to-back in 2010 of a series of papers that really bring together lines of evidence from evolutionary biology, statistical genetics, and molecular genetics, all of which implicate this gene, PRDM9, in specifying the location of recombination hotspots, at least in mice and in humans. So this gene can be represented by this cartoon here, where the 4 domains of the gene are shown. One of these domains, represented by this set of parallelograms, is a C2H2 zinc finger that binds DNA. And so each of the parallelograms here is one of the fingers in the zinc finger binding array. And 4 residues in each finger are in contact with 4 base pairs, 3 on the major groove, 1 on the minor groove. And because C2H2 zinc fingers, because the binding specificity of C2H2 zinc fingers is well predicted computationally, we get a reasonably accurate prediction from just generating this position weight matrix computationally shown here. And then below, you see a different representation of this zinc finger, where now each finger is represented by an oval, and the 4 letters that you see within each oval are the identity of the residues in contact with DNA. So it has a DNA binding domain. It also has a set domain that performs 2 epigenetic modifications. It trimethylates H3K4 and H3K36. And we now know that those 2 epigenetic modifications are actually causal in later recruiting the recombination machinery. And then it has these 2 other domains about which much less is known, other than the fact that they're lightly protein interaction domains. So the working model for how PRDM9 specifies the location of recombination hotspots is that it binds DNA, facilitated by its DNA binding motif, performs these epigenetic modifications, and then at a subset of sites that have been bound by PRDM9, the recombination machinery is somehow recruited. That includes SPO11. SPO11 comes, inflicts double-strand breaks on the genome, and it's the repair of those double-strand breaks that lead to crossovers and non-crossovers to recombination outcomes. So just to show you this in a little bit more detail, what happens then during this stage of meiosis is that the chromal axes, is that DNA loops form, along the chromosomal axes during meiosis. I see I have a typo on this slide, I apologize. And then PRDM9 comes and binds in these chromosomal loops, catalyzing then the formation of these epigenetic modifications. And then at these bound sites are thought to be tethered to the axis. And then somehow SPO11 is recruited to a subset of these PRDM9 binding motifs. In any given meiosis, a small subset, and initiates double-strand breaks. So the key point for our purposes is that PRDM9 marks the spot of future double-strand breaks, and it does so by binding to specific DNA sequences with greater affinity. And that property, the fact that PRDM9 binds some sequence motifs with greater affinity than others, in order to specify where double-strand breaks occur, has interesting evolutionary implications. And so the reason for that has to do with how the recombination, how double-strand breaks are repaired. Namely that when a double-strand break occurs on one of the homologs, that homolog then needs to copy the template provided from the template provided by the other homolog. And so if you have, imagine two homologs, a red and a blue, if a break occurs on the red homolog, you'll end up then with two blue alleles. If a break occurs on the blue homolog, you'll end up with two red alleles. And if blue and red homologs are equally likely to experience a break, half the time you end up with two blue, half the time you end up with two red. Nothing changes at the population level in terms of the frequency of blue and red alleles. If, however, one of the homologs is more likely to recruit PRDM9, because it has a PRDM9 binding motif, say, and the other one has a motif bound with less affinity, then what happens is that, say, this red allele is more likely to be cut, and therefore you're more likely to see two blue alleles at the end of the repair process. And this undertransmission of PRDM9 binding motifs in heterozygotes due to gene conversion actually acts in a way that is mathematically analogous to selection against the PRDM9 binding motif. And so very rapidly, you end up with the loss, at least in the absence of any countervailing force, you end up with the loss of this PRDM9 binding motif. So you can imagine then that this dynamic could potentially lead to the rapid erosion of specific hotspots, again, in the absence of any countervailing forces. Another property actually leads to the rapid loss of hotspots, and that is that the PRDM9 zinc finger itself is very rapidly evolving. So I've borrowed this figure from Simon Myers and colleagues to make this point. What they're showing here are the C2H2 zinc finger binding array, where again, each finger is shown here with an oval, and the identity of the residues in contact with DNA are the different letters. And you can see that not only are the residues in contact with DNA different, but actually the very number of fingers is different. And so there's this extremely rapid turnover of binding specificity of this protein across mammals. And importantly, this isn't just to do with the mutability of the sequence, because the rapid turnover of base pair is really concentrated at those residues in contact with DNA rather than throughout the zinc finger. In fact, the zinc finger as a whole is quite constrained. So why does the PRDM9 zinc finger evolve so rapidly? I'll return to that at the end of my talk, but for now I just want to emphasize that there are a number of explanations out there, but all of them link the rapid evolution of the PRDM9 zinc finger to its role in directing recombination. So putting these facts then together, if you imagine three hotspots shown in this cartoon, then if a cold allele were to arise at one of these hotspots, we would expect it to rapidly displace the hotter allele from the population, leading to losses of individual hotspots. And then we expect that through turnover of the PRDM9 zinc finger binding specificity, we're going to see gains and losses of whole new sets of hotspots. So that is the dynamic that we expect at species that carry PRDM9. What about species that don't carry PRDM9? Well, here there was a really interesting first hint from an experimental study in mice led by Galina Petukova. So what she did is she developed an assay which is kind of a variant of ChIP-seq on a recombination protein in order to obtain a quantitative readout of double-strand break frequencies in the mouse genome. And what she found is that mice that carry PRDM9 tend to make all their double-strand breaks not at transcription start sites as she defined them. And then she was able to knock out PRDM9, and she found, interestingly, that in that case, a much larger fraction of double-strand breaks occurred at or near transcription start sites. And the way that they interpreted these findings is that in the absence of the epigenetic modification, the trimethylation of H3K4 made by PRDM9, the recombination machinery is defaulting to the residual H3K4 trimethylation marks that it finds in the genome, and those tend to be associated with sites of transcription initiation. Now, consistent with these findings in mice, Adam Otten and Adam Boyko had a study where they looked at patterns of linkage disequilibrium in feral dogs and inferred population recombination rates using a population genetic approach. And dogs are of interest because, like other canids, they carry a pseudogene for PRDM9. And what they found is that in these dogs, like in knockout mice, recombination tended to be concentrated near transcription start sites, and specifically transcription start sites near CPG islands. So we were very interested by these findings and wanted to understand how general they might be, whether recombination really does tend to occur near promoter-like features in the absence of PRDM9, and most of interest to us, whether hotspots still evolve rapidly as they do, for example, in primates and in rodents, when in species that actually lack PRDM9. And so the work I'm about to tell you about was done by a former graduate student in my lab, Ellen Leffler, and a former postdoc in my lab, Sonal Singhal. So we were looking around for what species we might ask these questions in. And in that regard, we were guided by a paper that had been published by Harmit Malik and Chris Ponting's group that found that the PRDM9 gene was absent from the chicken genome. And so we surveyed the genomes of various bird species that had been made available at the time, and indeed did not find an ortholog of PRDM9, nor did we find an ortholog in the RNA-seq data we had for testes from a subset of these species. We didn't find it in crocodiles either. We did find an intact ortholog in turtles, and I'll return to that later. So on the basis of these observations, we decided to focus on a set of bird species, and specifically zebra finch, long-tail finch, and an outgroup double-barred finch. And we chose these species because, as shown in these boxes here, the nucleotide divergence between zebra finch and long-tail finch is roughly on par to what you would see between humans and chimpanzees. So that gave us a comparison to a timescale over which we knew that recombination hotspots are rapidly evolving in species that do have PRDM9. And for those of you who are interested, these gray lines here are 1,000 gene trees, and blue is the species trees, and the apparent incomplete lineage sorting you see is the result of the very large effective population sizes of these species rather than of gene flow between them. So what did we do? We sequenced 20 zebra finch and 20 long-tail finch, and so in so doing, obtained patterns of genetic variation throughout the genome. And then, as Adam Boyko and colleagues had done, we inferred population recombination rates from patterns of linkage disequilibrium, obviously separately in zebra finch and long-tail finch. So now we have genetic maps in the two species, and we can ask where recombination occurs and whether the hotspots are in the same locations in the two species. So in terms of where recombination occurs, we found that in these birds, as expected from the fact that they lack PRDM9, recombination rates tend to be increased near transcription start sites, and particularly near CPG islands. So what I'm showing you here is the fold recombination rate as a function of distance to the nearest promoter-like feature relative to the recombination rate 100 kb away. And I just want to kind of caution you against reading too much into the quantitative differences between species here, because these maps have different resolutions. But what does seem clear is that in species that lack PRDM9, recombination rates are elevated near promoter-like features. So what about the question of whether hotspots still evolve rapidly in these birds lacking PRDM9? So I've shown you this figure before. This is the comparison of human and chimpanzee. The degree of overlap we actually see in hotspot locations shown in blue here, which is several %, and barely more than what you would expect if you placed these hotspots more or less randomly along the genome. Now we can do the exact same analyses in zebra finch and long-tail finch, and the pattern that we see is very different. Where here, 70% of hotspots are observed as overlapping in their location in the two species. And because we don't have 100% power to detect hotspots by this approach, but we think it's very unlikely that we're detecting spurious hotspots independently in two species, we think this is a lower bound, and at least 70%, if not all hotspots, are shared between the two. So this was really intriguing to us, and we wanted to confirm these findings. To do so, we took advantage of the fact that recombination actually has two independent effects on patterns of genetic variation. On the one hand, it breaks down linkage disequilibrium, and that's the signal that we use to infer the presence of these hotspots in the first place. It also increases GC content by a process called GC bias gene conversion. So if you're not familiar with it, this is a process that, as work by Laurent Duret and Nicolas Galtier has documented, seems to be operating throughout vertebrates, that occurs during the repair of double-strand breaks. So when you have formation of heteroduplex DNA in the repair of double-strand breaks, if that heteroduplex DNA involves an A or a T on one strand and a G or a C on the other strand, then there's a bias towards repairing it to G or C. In fact, that bias can be quite strong. So Amy Williams and I, a few years back, estimated that in humans, that bias is about 2/3. So you're quite a bit more likely to repair towards GC. And the effect of that is basically analogous to selection for GC substitutions. And so you expect to see a flux towards GC substitutions where you see a breakdown of linkage disequilibrium. So we took advantage of that and we asked that question. We collated all the hotspots we had called on the basis of linkage disequilibrium data in zebrafinch and in longtail finch. And we asked, do we see excess flux towards GC in the lineage ancestral to our sample in zebrafinch and in longtail finch? And that's what you're seeing here. We see this elevation of GC star centered where we think the centers of the hotspots are, and we don't see them in cold spots that are matched for the same base composition, but where we don't see a breakdown of linkage disequilibrium. So that was reassuring to us. It also gave us an idea, which is that if we can see the footprint of recombination hotspots by looking for excess GC substitutions, then maybe we can see that even in species for which we haven't done population resequencing. And so we took advantage of that to query different bird genomes. And what we found is that there's at least circumstantial evidence for these hotspots still being in the same location, not just over millions of years that separate zebrafinch and longtail finch, but potentially tens of millions of years all the way out to collared flycatcher. So it seems that hotspots evolve rapidly in species with PRDM9, such as apes, and subsequent work has also shown rodent species, but doesn't evolve rapidly, at least in finches that lack PRDM9. And back to back with the publication of our paper came a paper from Scott Keeney's group in which he used sort of an approach similar to the one Galina Petukova had used and assayed the frequency of double-strand breaks in the yeast genome. In fact, in two very divergent yeast genomes. And he found that despite the 30% or so nucleotide divergence between these species, the vast majority of hotspots were in the same location. And in fact, not just were in the same location, but their intensities were conserved. Namely, they were used in similar proportions of meioses. So then that suggests a contrast, a link between how recombination is targeted to the genome and how hotspots evolve. Where when recombination hotspots are directed by PRDM9, they can be lost by the loss of individual binding sites and then gained or lost by changes to the PRDM9 zinc finger. And that model, and then if in species that lack PRDM9, instead what seems to happen is that recombination targets functional elements maybe associated with promoters, either because those functional elements are causal in recruiting the recombination machinery. So for example, CPG islands perhaps are causal in recruiting the recombination machinery, but don't disappear from the population due to gene conversion because there's selection to maintain them, because they play pleiotropic roles. Or maybe what's going on is simply that the recombination machinery is taking advantage of regions that are more accessible to it, and those happen to be associated with promoter features. Regardless, it appears that in these species, hotspots are much more conserved over fairly large evolutionary distances. So this is an interesting contrast, but at the moment when we completed this study, it was still pretty anecdotal. So we knew that apes and mice looked something like what we see to the left, and birds, perhaps dogs, yeast look something like what we see to the right. Of course, we were interested in understanding to what extent that pattern might be general. And so when I say we here, I now mean Zach Baker, a former graduate student in my lab who's now a postdoc with Guy Sella at Columbia, Molly Schumer, a former postdoc in my lab, and Yuki Haba, who was a master's student working in my lab. So together they decided to reconstruct the evolutionary history of PRDM9 in vertebrates and try to learn something about the generality of this sort of dichotomous pattern I showed you earlier. So the way that they did that is that they mined RefSeq for orthologs of PRDM9. And when they couldn't find it in RefSeq, looked in whole genome assemblies whether they could fish it out. And importantly, in all cases in which the gene model that they called for PRDM9 was sort of unexpected, they verified that they would get the same assembly by doing de novo RNA-seq assembly from transcriptome data, from testes data for that species. So every call that's made on the basis of a reference genome, which may be somewhat unreliable, is then backed up by this de novo assembly of RNA-seq data. In the end, they found 227 PRDM9 orthologs, and if you're still paying attention by this point, you'll notice that 227 orthologs is more than 225 species, and that's because PRDM9 has duplicated a number of times in vertebrates. So the results of their study are shown here, and the numbers in parentheses represent the number of genomes we were able to query for each of the species in each taxon. So the first thing you'll notice is that the intact version of PRDM9 known to direct recombination in placental mammals was already present at the origin of vertebrates. The second thing you'll notice is that there were numerous losses of PRDM9. So the loss in canids I alluded to earlier, the loss in birds and crocodiles that we discussed, also an apparent loss in amphibians, here on the basis of 3 genomes, but we've since extended that to a number of others. In addition to these complete losses of PRDM9, you'll notice partial losses of PRDM9, in particular, many instances where the species have lost the N-terminus domains. And again, we have an RNA-seq read that confirms that. So the next question we wanted to ask then is which of these orthologs of PRDM9 is directing recombination? Now, one question that you might have at this point is, well, maybe these partial orthologs are actually not functional. Maybe you're just spotting PRDM9 orthologs as they degenerate, as they become pseudogenes. And so the first thing we did is just check whether these partial orthologs were still functional by asking whether DNDS was significantly less than 1. And actually, in every one of these cases, it was significantly less than 1, indicating that they have some function. The next question we wanted to ask then is how can we identify the subset that might be directing recombination? And here we made an assumption, and it's just an assumption, which is that when PRDM9 directs recombination, its zinc finger is unusually rapidly evolving, as is the case in this handful of species in which we know independently that PRDM9 is directing recombination. So we just tested, are the residues on contact with DNA evolving unusually rapidly, where unusual is assessed empirically relative to the 100 or more other C2H2 zinc fingers in that species genome. And what we found was quite striking, and that is that we found rapid evolution of the zinc finger shown in green, if and only if we also saw the presence of the 2 N-terminus domains. And so we can ask how much of a coincidence that is. Specifically, we can test for whether those are unusually concordant, controlling for the shared evolutionary history of these species. And we find that indeed, this would be highly unlikely to, this partition would be highly unlikely to occur by chance. So it looks like rapid evolution of the zinc finger is tied to the presence of the N-terminus domains. And if rapid evolution of the zinc finger is indicative of a role in directing recombination, that implies then that the N-terminus domains must play a role in directing recombination. And one implication is that in species that don't have an intact version of PRDM9, that have, say, only this PRDM9 beta that lacks the N-terminus domains, we don't expect this ortholog to be directing recombination. So we decided to test that by availing ourselves of a system that Molly Schumer was studying in the lab of swordtail fish, specifically two species of swordtail fish, Xiphophora birchmani and Xiphophora malinche. And we availed ourselves of this system because these species hybridize naturally in multiple river systems in Mexico. And so what we can do is we can use them as kind of recombinant lines that are occurring in nature. We can sequence them at low coverage and infer where the ancestry switches occurred in the hybrids, and then learn from that where crossovers must have occurred in the previous 70 or so generations since these hybrids are thought to have formed. And so when we do that, we get a sort of fairly crude crossover map from these admixture patterns. And in addition to this genetic map, we also collected H3K4 trimethylation data from the testes of these species. So then we could ask, well, is the PRDM9 binding motif that we predict computationally associated with peaks of H3K4 trimethylation as expected if PRDM9 is responsible for these epigenetic modifications? We found it's not more than you'd expect by chance, taking into account the base composition of the motif. We can ask, is the PRDM9 binding motif associated with increased recombination? And we find that it's not. And finally, we can ask, where is recombination occurring in this species? And we see that, as is the case in species that lack PRDM9 altogether, it seems to be somewhat elevated near promoter-like features. So at least based on this one example, it does indeed seem that partial orthologs of PRDM9 are not directing recombination. And in fact, luckily for us, in some ways, after publication of our paper, there were two studies in mice that knocked out specifically the crab domain and showed that a PRDM9 that lacks the crab domain only behaves essentially like a knockout, as if it were lacking the gene altogether, in terms of its recombination phenotypes. Now, that's one implication of our finding. The converse, of course, implication is that if you have a complete version of PRDM9 and the zinc finger is rapidly evolving, then PRDM9 does direct recombination. And that should be true, therefore, not only in mammals, but also outside of mammals in species like snakes and lizards and turtles. And so we're in the process of testing this using various sources of genomic data in corn snakes in collaboration with Athanasia Zika at the University of Geneva. But actually, a couple months ago, Todd Casto's group published a paper in rattlesnakes where they were only able to obtain a few of the zinc fingers of the zinc finger binding array, but where it really looks like a population recombination rate, increases close to the predicted PRDM9 binding site, indicative of the fact that PRDM9 is in fact directing recombination rates in this snake. So putting this together then, it seems like there are at least two strategies for directing recombination in vertebrates and possibly beyond, where either recombination hotspots are directed by a complete PRDM9 with all four domains I showed you previously. And in that case, we would expect the recombination landscape to evolve very rapidly, both due to these gene conversion against hotter alleles and due to changes in the PRDM9 zinc finger itself. And that model seems to apply to primates, rodents, potentially snakes, and by extension then also turtles and some fish. Or you lack a complete PRDM9, either you lack PRDM9 altogether or the species lacks the N-terminus domains. And in that case, it looks like recombination is occurring primarily, although not exclusively, at promoter-like features, and for reasons that remain to be understood, are much better conserved over evolutionary time. And this model seems to apply to birds, canids, some fish, and again, by extension, maybe amphibians and monotremes. So what's interesting then with these sort of two main strategies in mind is to go back to the phylogeny, because what's quite clear is that these strategies are then quite interdigitated across vertebrates. And that raises a number of different questions, right? So for one, we see that PRDM9 was somehow adopted early on before the origin of vertebrates and conserved over hundreds of millions, billions of years, and yet repeatedly lost. So an obvious question is, well, why adopt PRDM9 in the first place and why keep it? So I don't know is the short answer, but I'm going to speculate. And I'm going to take a step back in speculating to why we might have hotspots in the first place. So one possibility that seems plausible to me, and I should say was greatly informed by discussions with Zach Baker when he was in my lab, is that the reason that there are hotspots at all is that they reduce the state space of the homology search, right? In other words, that we have hotspots to make homology search more efficient, that when you're trying to align the two homologs along their length, you only need to check a subset of the chromosome for homology. And a nice kind of acute observation that's at least consistent with this idea is that two taxa, I should say, known not to have these recombination hotspots of 1 to 2 kb are two sets of organisms that don't depend on the homology search to synapse. That is, in Drosophila and Caenorhabidopsis, the chromosomes zipper up and then they recombine. They don't use recombination for the homologs to find one another. And so that's consistent with the view that if you're not dependent on recombination in order to synapse, then you don't need hotspots, and therefore hotspots are probably related to this homology search. And I should say that this isn't my original insight. In fact, it's something that Scott Hawley pointed out to me many years ago when I was a graduate student, and so I'm sort of playing very slow catch up on that idea. But assuming it's true, then maybe the advantage of using PRDM9 is to make the homology search even more efficient. Because if the distribution of binding affinities looks something like this, as seems plausible, then PRDM9 binds to a small fraction of the genome, and there's a much smaller state space to explore to find your homolog. So in that sense, in the same way as the existence of hotspots helps to reduce the state space, the existence of PRDM9 helps to reduce it even further. Okay, so let's imagine that's why PRDM9 was adopted in the first place. How could it be then lost so readily? How costly is it to lose PRDM9? So the answer to that, interestingly, seems to depend somewhat on the species. So if you knock out PRDM9 in most mice strains, not all, but most, males are sterile and females have somewhat decreased fertility. In contrast, dogs and canids more generally carry a pseudogene for PRDM9 and are obviously not sterile. In humans, we also have an interesting clue about that comes from a study that looked at the offspring of consanguineous matings and found a woman who carried 2 loss-of-function variants of PRDM9 identical by descent, but had 3 children. So we know that in humans, at least in human females, the loss of PRDM9 does not lead to complete sterility. But of course, complete sterility is a very large selection coefficient, and it remains possible that there's weaker, even quite strong selection operating on loss of PRDM9 that isn't a selection coefficient of 1. So the question remains of whether PRDM9 might just be dispensable in some species. In other words, whether its loss is simply neutral. And interestingly, that's a question we can actually address in humans. We can ask whether the loss of PRDM9 is likely to be neutral. And we can do that using a population genetic approach. So specifically, what we did is to consider a huge data set of a couple 100,000 exomes that were collated by the Broad Institute, this data set NOMAD. And the individuals that were resequenced are not sterile and are alive, so we can use the genetic variation seen among them in PRDM9 to gain a sense of what can be tolerated. So specifically, what we do is we look at the observed frequency of loss-of-function variants in this gene, and then we ask how likely this observed frequency of loss-of-function variants is given a mutation rate we take from pedigree studies, a plausible demographic model, and a given selection coefficient. And so we can estimate the posterior distribution of the selection coefficient given these data. And what we find is that the mode of the posterior distribution is a selection coefficient in heterozygotes of 1.5%. So the most likely selection coefficient that, given the data, is something between 1 or 2%. But most importantly, there's very little probability mass around 0, suggesting that a single loss-of-function variant of PRDM9 is actually unlikely to be neutral. So PRDM9 may not cause sterility in women, but losing it doesn't seem to be costless either, at least not in humans, and we know not in mice since it causes sterility. So how then can PRDM9 be lost? So again, I don't know, but increasingly I suspect that it's linked to the answer to another question. And that question is, what drives the rapid evolution of the PRDM9 zinc finger? So I'm going to try to answer that question and then return to the question of how it may be lost. Now, in answering this question, I'm leaning heavily on work that was done by Zach Baker, in which I'm involved, but is primarily his work in collaboration with my colleague Guy Sella, with whom he's doing a postdoc. So their model is based on recent discoveries about a second role of PRDM9. Now, recall that SPO11 makes double-strand breaks, and then obviously these need to get repaired. So as it turns out, there's now pretty strong evidence that this repair is much more efficient and more likely to lead to a crossover when PRDM9 has bound both homologs, rather than when it's bound only one. So PRDM9 increases the efficiency of repair of double-strand breaks when bound on both homologs. And instead, the double-strand break repair is delayed when it's bound on only one. Why that is not known exactly, but one possibility supported by recent results from Scott Keeney's lab is that what PRDM9 is doing is helping to reposition chromatin on the uncut chromosome, and therefore helping the strand invasion that's needed to find a template for double-strand break repair. So the key here that I want you to remember are not these details, but that symmetric binding on both homologs is good. It enables more efficient repair of double-strand breaks. So why does all this matter to PRDM9 evolution? Well, for a given amount of PRDM9, if there are few binding sites used, they're going to tend to be bound symmetrically. But gene conversion we saw of hot to less hot binding sites leads to the loss of PRDM9 binding sites, and in particular, the loss of the hottest sites, which are cut more often by definition. So conversion then from hot to lukewarm binding sites leads to the extinction of the hottest hotspots. And by leading to extinction of the hottest hotspots, it leads to an increase in the number of sites in the genome that can be bound. The consequences of increasing the number of sites bound is that fewer are bound symmetrically. That's not good because it slows down the efficient repair of double-strand breaks. And if extreme, if in fact most double-strand breaks are asymmetrically bound by PRDM9, it can lead to asynapsis and infertility, as has been documented in hybrid mice. So what Guy and Zach's model suggests is that that's the source for the selection on new PRDM9 alleles. The reason that the PRDM9 zinc finger is so rapidly evolving is that there's recurrent selection for new PRDM9 zinc fingers as a result of selection to retain symmetrically bound hotspots. So there are many models out there that posit that the zinc finger of PRDM9 is rapidly evolving to keep as many hotspots as possible in the population. This is instead positing that it's not about the number of hotspots, but the number of symmetrically bound hotspots. So I've speculated about why PRDM9 may have been adopted, namely to enable efficient homology searching. And I also speculated, based on Zach and Guy's model, as to why PRDM9 binding is rapidly evolving, namely to retain symmetrically bound hotspots and keep the repair of double-strand breaks efficient. And potentially this same dynamic could also allow for loss of PRDM9. It could be that under some conditions, not using PRDM9, say using promoter regions instead, is good enough in that enough hotspots are bound symmetrically in most meioses. And if so, PRDM9 will no longer be as useful, and its loss may not be that costly and potentially can be overcome by genetic drift. So, of course, there are numerous other questions raised by this phylogeny. For example, what about the partial orthologs of PRDM9? What are they doing if not directing recombination? And in that regard, there are some interesting hints that in teleos, these PRDM9 beta are all evolving in parallel towards some potentially new similar function, possibly as readers rather than writers of epigenetic marks. Another set of questions one might ask is, why did PRDM9 duplicate so often, and why were these duplicates conserved? And again, we don't know the answer to that, nor do we know, for example, how often the duplicates are both involved in recombination. So I'm going to leave you on these open questions, but I want to take a few minutes to acknowledge the people whose work I was able to present today. So first and foremost, Zach Baker, who conducted the work that I showed you on the phylogenetic history of PRDM9 and the model on which I based myself at the end and informed a lot of my recent thinking about this topic, together with Molly Schumer, again, a former postdoc in my lab who's now a faculty member at Stanford. The bird analyses were done by Ellen Leffler, who's now a faculty member at University of Utah, and Sonal Singhal. Then the work I showed you on inferring fitness effects of loss of function variants in PRDM9 was done by my current postdoc, Zach Fuller, who's actually applied this kind of analysis to all the genes in the human genome. Carla Hodge is working on patterns of recombination outside of mammals. And I didn't have time to show you work by Isabel looking for the coevolution of PRDM9 and other meiosis genes across vertebrates. So maybe I can reappear now and answer any questions. Thank you very much.
Great. So thank you very much, Molly, for a fascinating and enlightening talk. That was really interesting. So we have quite a number of questions for you on our Slack channel. And I also had one question I also wanted to ask you before we kick off with the other questions. So I'm quite interested in whether you see any sex differences at all in patterns of recombination hotspots, and also specifically in relation to PRDM9 and its links you were talking about to double-stranded DNA repair. If you could tell us a bit more about that, please.
Yeah, so in terms of hotspot usage, this has been looked at quite carefully in humans by Adam Otten in particular, and it seems like there are some differences in hotspot usage between males and females, but most hotspots are used by both sexes. In terms, however, of the consequences of asymmetric binding of PRDM9 on fertility, there do seem to be marked differences between males and females. So there are much stronger sterility effects in males than in females. And that might be because there's a more stringent checkpoint for meiosis. It might be because of the bridges in spermatogenesis leading to a nonlinear effect, where if some fraction of sperm are not viable, then all the sperm are killed off, which isn't the case in egg development. So we don't really know why that is, but it's definitely the case that the fertility effects are stronger in males than in females.
Okay, thank you. That's really interesting. So we now have some questions as well in our Slack channel. So we'll start with a question from Julie Blomart. So she asks, did you see any relationship, negative or positive, between these conserved hotspots in birds and the number of transposons in those areas?
So the short answer is that we didn't look for that specifically, so I can't answer that. A slightly broader answer is that we're actually very interested in understanding determinants of hotspots in species that lack PRDM9, specifically hotspots that might be away from promoters, because not all hotspots are occurring at promoters, even though there's an enrichment near promoters. And so we'd like to understand what those features are, and also if there are a subset of hotspots that aren't conserved in those species, what's driving their evolution. So we know in yeast, for example, that rearrangements can lead to shifts in hotspots between species, chromosomal rearrangements, but we don't have that kind of, to my knowledge, we don't have that kind of information for vertebrates.
Okay, that's great. Thank you. So the next question we'll move on to is from Judith Mank. So you talked about this a bit towards the end of your talk, but she asks, given the importance of recombination and the broad conservation of PRDM9 in vertebrates, do you have any ideas about why PRDM9 has been repeatedly lost in some lineages?
Yeah, so this is complete speculation, but what I think, sort of informed by Zach's results, is that it's possible that the important feature of PRDM9 or an important feature of PRDM9 is to have a sufficient number of double-strand breaks bound symmetrically, especially on small chromosomes, because one feature I didn't actually go into is that because there's a delay in the repair of double-strand breaks that are not bound symmetrically, the ones that are not bound symmetrically are less likely to become a crossover. And you need crossovers in order to ensure the proper segregation of chromosomes to the daughter cells. And so you could imagine that if you don't have a sufficient number of symmetrically bound hotspots on, say, short chromosomes, then that endangers fertility. And so the idea would be, well, maybe you lose PRDM9 when you can accomplish that by other means. For example, if you're only using a subset of promoters, 1 in every 10 promoters, maybe that's enough symmetrically bound hotspots as it is that you don't really need PRDM9. And so the loss of PRDM9 is not that costly. So maybe the question is about the competition between PRDM9 directed recombination and then this residual mechanism that targets promoters, and if that residual mechanism is good enough to ensure a sufficient number of symmetrically bound hotspots, then it's not that costly to lose PRDM9.
Yeah, I see. So a balance between those two. So we'll now move on to the next question. So we have a question. Is there any relationship between PRDM9 conservation and genome size? And could it be more important in very large genomes?
So I don't think so is the answer to that question. We looked across vertebrates at whether there were kind of features like genome length, fraction of total genome that is repetitive, fraction that are classified as certain transposable elements. And we didn't find any clear correspondence of presence, absence of PRDM9 with those features. Also, I suspect that, that the total genetic map might matter more than the total physical length of the genome. So another corollary of Zach's model is that it's possible that if you make enough double strand breaks, that might allow PRDM9 to be lost because you're making enough of, I mean, crudely, because you're making enough of them that a large enough number will become crossovers. And so in that sense, it might be the total genetic map in centimorgans rather than in base pairs that matters.
I see. Yeah, that makes sense. And related to that, could the levels of methylation be linked with the number of PRDM9 duplicates?
Don't know.
Okay. Great. So we have another question. There's a question about polyploid organisms. So the question is, how does recombination work on polyploid organisms? Are hotspots more frequent? And is there a mechanism, a PRDM9 mechanism present on them?
Yeah, so this is not something I know much about, but Kristin Bombliss has done work, really nice work, looking specifically at this question and how polyploidization copes with the tangles that are induced by recombination. And so some of these species definitely have hotspots. Plants have hotspots. They don't have PRDM9. And the more general question, I guess, point is that it's complicated because if you have more than one crossover per chromosome, then because you can pull different chromatids into each crossover, you end up with these bundles, these knots that then endanger proper segregation. And this is all quoting Kristin's work, nothing to do with my own. And my understanding, but this is secondhand, is that basically one way to reduce that problem is basically to decrease the number of crossovers or increase the strength of crossover interference to reduce that tangle problem.
Okay, that's great. Thank you. I think we'll go with one more question for the time being, and then perhaps if you're happy to go through some of the questions in the Slack channel afterwards, and
Sure.
maybe answer some of the ones we haven't had the chance to get onto today. So the final question we're finished with today. So there's a question, is PRDM9, does it also have a similar role in meiotic recombination?
No, to my knowledge, at least none that's been documented. And in fact, in some species, it's only expressed at low levels in meiotic prophase. So that's one of the lines of evidence suggesting that whatever is driving its rapid evolution is almost certainly at least linked to that stage of meiosis, if not to its role in recombination.
Okay, that's great. Thank you. So yeah, that's all we have time for today, but it is our great pleasure to thank Molly once again for a truly fascinating talk and a great launch to our Evolution and Ecology seminar series. Thank you very much, Molly.
Thank you, and thanks again for doing this.
Thank you. So a reminder to everyone that our next talk coming up in the seminar series will be from Professor Hamid Malik, from Fred Hutchinson Cancer Center in the USA, on the interesting topic of arms races between host and viral genomes. So this talk will be taking place on Friday this week, at the same time and same place, and we hope that you can join us for this. So in the meantime, check our updates on Slack and on our Twitter feed. Thank you for joining us, and until next time, stay safe, see you again, and goodbye. Thank you very much.